They say hunting and fishing can be dated back more than 2 million years. Throughout the years, the historic angler and hunter has steadily evolved into the avid hunter and angler that we know and love today. Welcome to the Outdoor Defiance podcast. As you listen, you will hear comical stories, companies talking about their products, researchers talking about their research, and overall ways to help you become a better outdoorsman. Welcome to today's episode.
What's up, everybody? In today's episode, we get to talk to the amazing Dr. Krysten Schuler about CWD and other disease impacts and disease ecology that happen to our white-tailed deer populations. CWD is a tricky disease. It's a real nasty disease, and we kind of dive into the different things to look for, what the disease looks like, different ways to mitigate it, and we also talk about different things such as the research that Krysten has done. It's pretty amazing where we started and where we are now. And it's interesting to listen to her talk about where we're headed. This is something that's going to impact us here in Oklahoma, I'm afraid, sooner rather than later. And it's already impacting much of the U.S. So it is definitely something we need to be on the lookout for, and definitely something that would be very beneficial to learn about. So without further ado, enjoy the awesome Dr. Krysten Schuler.
Krysten, thank you for coming on today's episode. I'm excited to talk about CWD and some of your other studies you've done. But before we jump into it, let's kind of start with the generic question we start with and give us a small background of, where you decided, all right, I'm going to study this, or, did you get started in hunting, and kind of what blew up this whole thing of your whole background?
Sure. Yeah, I'd be happy to. Most people think that there's like a clear path and you knew what you wanted to do. And mine was sort of more of a meandering series of opportunities. So I did not, full disclosure, I did not grow up as a hunter. I grew up in Northwest Pennsylvania, like right in the middle of the national forest. I rode horses and everything. But my dad was, really big into hunting and fishing, trap shooting, all those types of things. And at the time, I didn't really, get it. I was sort of opposed to killing animals, but of course ate venison like all the time, and fish and everything like that. And I went to school for biology, doing my undergrad, and it was almost like a series of things I didn't want to do. Like I didn't want to be a doctor. I didn't want to be a veterinarian. Like I didn't want to work with people. But I really wanted to work with animals. And so like people didn't really understand. They're like, okay, so you're going to be a park ranger? Like, no, there's got to be something else. And so I graduated with no job and sort of no plan, and ended up working in a lab that was pretty boring. And during that time, actually found out about like wildlife management by going to the library and looking at the Journal of Wildlife Management, Journal of Wildlife Diseases, things like that, and managed to apply for a master's program at Oklahoma State. Go Pokes. And I was going to work on bison. So I got offered a master's working on deer in Nebraska, but it was kind of one of those things. I'm like, everybody works on deer. Like bison sound really cool. So let's go and do that. And while I was at OSU, I took a class on wildlife diseases, and, just thought it was super interesting, and decided I wanted to do a PhD, and had the opportunity to sort of choose between two options. One was working on elk in Yellowstone, which would have been totally amazing. But the other one on chronic wasting disease in Wind Cave National Park in South Dakota. And so I chose the disease path instead.
Wow.
That got me on this road.
So when you went up to you said South Dakota, correct?
Mm-hmm.
Did you have like a big idea of what CWD was, or was it kind of a smaller idea?
Yeah, I think at the time CWD was still pretty small, because this was, early 2002. And at the time, CWD was something that had just been kind of seen in Colorado and Wyoming. It was moving a little bit into Nebraska, and South Dakota was just discovering its first cases. So it was really still, not on people's radars. And then it was that fall 2002 hunting season when Wisconsin found it that everything sort of blew up, and it became a much bigger deal since that was the first detection east of the Mississippi River. And that's, sort of the explosion that everybody started paying attention to.
Yeah, I feel like that's kind of where it's at in Oklahoma. It's like everyone knows it. It's like kind of on the radar, but it's not. They just kind of blow it off because it's never really affected us that much yet. So it's kind of
the general public, people like us, we talk about it. I'm terrified of it. I'm not gonna lie. I don't know. You just don't know how it's going to affect everything. I mean, you kind of do, but
you're gonna have to tell me if you're more or less scared after we talk about it. We'll gauge, feel your baseline right now, and then after we're done, see how you feel.
Okay. Well, yeah. We got to talk to Dallas Barber, and these wildlife agencies, they have plans, but I mean, we don't know. You know what I mean? Mm-hmm. Wisconsin, you hear about that case study, and that's kind of the poster child of what not to do. It seems like in our classes, that's a lot about what they talked about.
Oh yeah.
But yeah.
So after I graduated from South Dakota State, I got a job at the U.S. Geological Survey's National Wildlife Health Center in Madison, Wisconsin. So I was, right there sort of in ground zero, seeing what was happening. And the management that they tried to apply, was pretty innovative at the time with Ernebuck. But everything just went sideways for the Wisconsin DNR. That was CWD partially led to a new governor being elected. The DNR lost control of a lot of different things because of CWD, because there was such a backlash against people. There was a really kind of twisted communication issue where the Department of Natural Resources said, we want to eliminate the disease. And in the public's mind, they heard, we want to eliminate deer. And obviously, in Wisconsin, deer hunting is, next to church. So that really, the public, really pushed back hard on that. And, the agency couldn't do really any major management afterwards. So studies are coming out now. I think what you mentioned, Gunnar, from a sort of a longer term collaring study where they're seeing population declines in areas that have had the disease the longest. So they haven't finished publishing those papers yet, but they've presented it to their commission where it's documented, so they know that CWD was there before 2002. They're not quite sure how long, but in that, we'll say, at least known of for 23 years, it's already sort of tanking the population in some of those counties.
I was going to say, before we kind of get into the nitty gritty and the, I guess, deeper questions, for people that don't know, can you kind of go into a brief generalization or a summary of what CWD is?
Absolutely. So CWD, or chronic wasting disease, it's named that because of what it does to cervids. So members of the deer family, mule deer, white-tailed deer, elk, moose are affected, but not as frequently. Reindeer and caribou can be naturally infected, and a few other species. There's some species of cervids that don't seem to be naturally susceptible. But it's caused by a protein. So it is really unique because it's not a virus, it's not a bacteria. And this is a protein that we have in our bodies naturally. And for some reason, which is still unknown, it changes its shape. So the normal protein in our body exists throughout in different cells. But when it changes shape, the mechanisms that our body uses to kind of clean out those old proteins, which are called proteases, go away. They can't bind to the abnormal form anymore. So if you think of it as like a lock and a key, and then all of a sudden the key changes shape and doesn't go in the lock anymore. So these proteins start building up and they form plaques on the surface of brain cells. So very similar to what they're seeing now with diseases like Alzheimer's and dementia. And these plaques essentially smother that brain cell. And when the brain cell gets smothered, it dies. And that leads to holes in the brain. So there's other diseases that are in the same family, and they're known as transmissible spongiform encephalopathies. Transmissible because it can go to animal, spongiform because literally the brain ends up looking like a sponge because there's holes in it. So CWD is the only one known in free living animals. The most famous one is mad cow disease. So that's what most people are familiar with.
So real quick, I want to say, I told Gunnar before we got on here, I was like, hey, I know you've had these classes and you've heard these big words. Like, you're going to have to ask some questions that I don't know. And you said you want to know our baseline before this. And I think I think I have a good baseline for this episode because I'm going to be kind of like the Oklahoma average hunter, like I know about it. I know how it's spread. I know how it works. Actually, some people probably don't even know that far, honestly. But after that, it's like I've heard about it and what it can do, but I've never seen it. So so that's my baseline. So I wanted to throw that in here before he talked, and afterwards I'll tell you like, oh no, or so. But so with CWD, in the baseline I have, what like when an animal gets it, what do they start doing? Like, what are the first symptoms and signs?
Yeah, that is a great question. They are fine when they first, so they'll ingest an abnormal prion, and that starts making their normal prions kind of like crystals forming, makes their normal prions change to that abnormal shape. And so then, their body's still making normal prions, they change shape and start multiplying. So for a year or two, they're totally fine. Nothing, major is happening. And it's towards the end stages when those plaques on the brain cells really start forming and they start getting a lot of holes in their brain. That's when you see abnormal behaviors. So that's when they start, if you have holes in your brains, you're not going to be normal. Like you can't function normally. So like you said, you've never seen it. It's hard to tell the difference. Like I deal with a lot of different deer diseases, and you have deer acting abnormally for a variety of reasons. It may be a bacteria pathogen, it may be trauma. Like we don't know until we get our hands on animals. So that's why it's really important for the members of the public, hunters, when they see something abnormal, to call it into their state natural resource agency. But at that end stages, when they've got holes in their brains, people may see, they're really skinny, like their hip bones are sticking out, their ribs are sticking out. They might be drooling, their ears can be down. They might not be aware of people. They might be hanging out around a house or another structure. And sort of the really end stages when their body just isn't functioning anymore. A lot of times we'll see on necropsy, which is the equivalent of an autopsy on an animal, aspiration pneumonia. So they're trying to drink because they pee a lot and they're drooling, but their throat doesn't swallow right. So they actually swallow water into their lungs, and that's not good for your lungs and can die from that. But like I said, living normally for a year plus, they're walking around just fine. But during that time, they're shedding prions in their saliva, their feces, and their urine. So prions are nasty little buggers because they're not broken down by the normal things. Like we think about, viruses can't really live outside a host for very long, or bacteria don't usually last that long. These things can last for years in the soil. They can be taken up into plants. So, a deer is alive for a long time, shedding a lot of prions. And then when they die, all those prions that are in the body, the highest concentration is in the brain, but that carcass decomposes on that spot, and that creates a hot spot where there's a whole bunch of prions. So more plants growing up there can have prions in them as well.
So I think a lot of people get CWD and EHD confused. Can you
Yeah.
kind of tell us the differences of that real quick early on?
Yeah, you're absolutely right, because hunters are like, I've seen EHD before, I've never seen CWD.
Mhm.
And not seeing CWD is a good thing. Um, but the prevalence of CWD, even where it occurs, is generally very low. Um, so when you might see dead deer from EHD, it's likely going to be in the summer when it's hot. And EHD is epizootic hemorrhagic disease. It's caused by a virus that's transmitted by biting midges or no-see-ums. Their species is Culicoides. And so what happens is these midges, bite an infected animal, they take a blood from it, they go and bite another animal and inject that virus. After a deer is infected, it doesn't necessarily die. EHD kills a lot of deer, but not all of them. If a deer gets really sick, it might die in like 5 to 7 days. And usually people will see them around water sources. And that's because they get a really high fever. And so they're going to water to try to, drink or cool off. And then they die in short order. But it's a completely different disease. So with EHD, you might see a lot of dead deer very quickly, but EHD doesn't happen every single year. It doesn't necessarily always get worse, where CWD typically always gets worse. So you might have a die-off from EHD, but then the deer population is able to come back from that relatively quickly. So, and that's usually like localized pockets as well. So you might get immigration into that area.
Can a deer survive EHD?
Yeah. Yeah.
And then,
So, oh, go ahead.
I was gonna say, can they survive CWD?
No,
No.
there's
Okay.
been no known cases of surviving CWD.
Well,
And we can talk a little bit about, genetics are a big thing, especially in Oklahoma right now. So we can
Yep.
talk about that if you want to.
yeah,
Yeah.
absolutely.
Before we get into the genetics, I heard you kind of mention the prions. They can get in the soil and they're prevalent for a long time. I skimmed over a paper that you were a part of today and talked about machine learning or modeling.
Mm-hmm.
And it talked about different variables that might increase the likelihood of spread. What type of ecotypes that you've seen kind of favor, like give it like a favorable spread, I guess, to the contagion?
Yeah. So if prions weren't so scary, they're really fascinating, things, because it's a protein, so it's not alive, but it does have a charge to it.
Mm-hmm.
And so there's certain soil types like clay that also has a charge. And so these prions will bind to clay particles and they actually become more infectious. So if we look at, the landscape and where prions are likely to stick in the soil and kind of remain on the surface, where like sandy soil, they might get washed down below the surface level. That is something that I think we need to consider. So the paper you're mentioning, we looked at all sorts of different factors like distance to rivers and things like that, thinking about migration corridors and animal movement. And our predictors weren't spectacular, but there's some other researchers that just redid that analysis using some different machine learning techniques, and they got it pretty good with predicting the likelihood of CWD emergence, but he hasn't gotten, he just did it. So I don't know if they've gotten to which predictors are yet. So, definitely higher deer densities can help promote transmission, just, increased contact between animals. It's really interesting because males typically have higher prevalence rates in areas, starting out in the beginning of the outbreak. If you think about a buck during rut, it's going around to a bunch of does, it's fighting with other males, there's a lot of interaction among deer there. And then during the non-rut season, if they're hanging out in bachelor groups, sharing pathogens and whatnot, versus, a doe that might be having a fawn and she sort of isolates herself. but then they come together in bigger groups during the winter. So all of those kind of play into how these diseases get transmitted as well. And if you have more deer in a smaller area, then that promotes transmission.
So you said genetics. Is there genetics in deer that can survive more, or no? I guess surviving more is not the right way, because you said if they get it, they don't survive. But is there deer that are more like acceptable to it, or?
Well, that's a question that's a really hot area of research right now. So there are certain genotypes that seem to sort of get diseased faster. So they die sooner when they're infected in experimental trials. So these other deer still end up dying, but it takes them longer. So there's sort of a debate, like, is that better? Because then, they survive another year, they can have another crop of fawns to kind of keep the population up. Or is it worse because they survive longer, and during that time they're shedding more prions into the environment? And there's a big question now in captive cervids about these different genetics, and if that could kind of sustain a captive herd and not let them get diseased. In Oklahoma, there was a bill introduced to release some of these deer that are supposed to be more genetically resistant. But the problem that we have is they haven't done the challenge trials yet to see if they actually survive it. Like, the study is all based, again, on machine learning, which is great to form a hypothesis, but then you'd have to go ahead and test and see if it actually holds up. So if you're following the scientific process. So I think we have more to learn on that, but I really hate putting all our eggs in one basket and being like, this thing's the one that's going to save us, because the other side of the coin on this one is that these deer with different genetics, there's also the possibility of having different strains of prions. And so I don't know how, the people who are really into prion strains can argue, if there's 9, if there's 20, if there's more than that, I don't know. But if you have different, deer genetics, these prions seem to adapt to their host. And so they might change the pathogenicity. So like the ability to infect and kill things. If you breed, a different genetic deer, then it might have a different prion genetic strain that might be more lethal to humans. And that kind of brings up the question, have there been any humans? And the answer is no. So that's good news for us so far.
See, I didn't even think about that angle. Thanks. I'm gonna.
I know this is just like it's like a spider web of problems.
See, my angle when I first read it, I thought kind of along that side of thinking of, well, if it lives longer, it's just gonna, it's just gonna drop off more, those prions. This was my first thinking. I didn't think about the other side of the coin where you said they might die earlier. That's an interesting thought. I hadn't thought about that. Yeah, that's scary. Thinking, that's just what, diseases in a sense do anyway. They adapt.
Yeah, right. Like, we can't be naive and think that, the prion's not going to change too.
Yeah.
Ooh. Yeah, it would be something like COVID where we're worried about the beginning and now we're kind of a little looser on stuff, or is it going to get worse? That's kind of all the diseases
So what
are.
is your overall thought on what what Oklahoma proposed? Like we kind of talked to Dallas about it, and he was like, I don't want to say a whole lot until I see what, the research, says, like what aren't we tasking what Texas A&M with this research? Isn't that what we're doing?
Yeah. But that's the problem. There hasn't been any replication, right? Like, how you can use whatever euphemism you want to use, like put all your eggs in one basket or let the horse out of the barn. Like, I'm all for testing it out and seeing how, these deer that are supposed to be, sort of genetically superior and resist prion infection, how they do. I mean, set up a research study and dose them and see what happens. Like that seems to be the next logical step. And I know that there's some captive deer owners that have had some of these animals that are supposedly have, really good genetic values, that have had CWD. So there's been anecdotal evidence kind of on both sides. So it's hard for me to judge without having, the controlled studies to really get at it. But I just think releasing animals before we complete that research, like you can't get them back. Like that's
Yeah.
it,
That's
I
what,
mean, yeah.
when it was released, it was like, what genetics are we looking at? Are we looking at on their head? Or are we looking at what they can do against CWD? And that's what, a lot of
Yeah.
people were thinking was, well, are they really trying to aim for CWD, or are we trying to look for the
Yeah.
rack? And that's kind of one of my worries, was because I don't know, a lot of people pushing it had deer farms, and some people pushing it have guide companies here. So it's like, I could see their side of the fight too. So yeah, I definitely think they need to research a little more before we just start letting random deer out. Because
Yeah. And fortunately, I think the majority of people who own deer, are want to see that research done as well. I mean, it's awful if they get a CWD diagnosis. I participated in a depopulation of red deer in New York this past fall, and it's horrendous for those families. Like, I'm not downplaying what they go through at all. And these people were doing everything right. They had a closed herd, but we haven't been able to get any funding to try to figure out where that infection actually came from initially. So it's really difficult to make progress on this disease. I know that there's people out there saying, "Oh, researchers are just making money off of this," and it, at least for me, it's not going into my pocket. Like, that's not really how this type of research really goes.
yeah, well, it's also devastating to the, like you said, the families. Like there was a lady in what is it, Cushing, that had a elk ranch here, and they raised world class elk here. As a kid, I went there, and I kind of forgot about it. And then I guess she got, I don't know if it's CWD, I don't remember exactly what it was, but they just came through and wiped everything out.
Yeah.
And like she couldn't sell any of her troughs for like 10 years unless she boiled them in water. It was insane. Like they literally took this lady's livelihood and just out the door in a day.
Yeah. Well, I mean, fortunately, there typically is an indemnity program. I mean, they don't pay the owner the value of those animals, but they do recoup something. And it's really unfortunate, but depopulating is the best solution that we have, and it's used for almost every other disease. So you think about like highly pathogenic avian influenza right now. Like if there's a poultry farm that gets that, like all those chickens and turkeys are gone. Like they've killed millions and millions of them across the country because of that disease. That's the same thing that happened with mad cow in Europe when they had it. They killed millions of cows to stop that disease. And even here in the US, we don't talk about it a lot. People, scrapie is a similar disease to CWD in sheep and goats. And people say, "Oh, we bred our way out of scrapie." Yes, there are some sheep that are resistant to scrapie. There is potential they could get an atypical form of scrapie, but they also killed millions of sheep too. So unfortunately, depopulation is just like the way that it goes for a lot of these animal diseases. We just don't have the vaccine or the treatments available that we do for people.
Let's take a break from this podcast and talk about some awesome products.
Hey folks, let me tell you about GritKnives. They're not just your average knife company. They're all about high quality, affordable, and unique products that'll make your outdoor adventures legendary. From limited edition pocket knives to rugged fixed blades and precision scalpels, they've got it all. Plus, their logo wear will have you repping the brand wherever you go. So whether you're out in the field or tackling everyday tasks, trust GritKnives to be your go-to for top-notch gear that won't break the bank.
Are you looking for some of the best sounding turkey calls on the market? Then look no farther than JAG Calls. Since 2022, Team OD has been using their calls and have had great success doing so. Two of my personal favorite calls are the Mother Clucker and the America. And I mean, if it has a name like that, it must be good. Check out JAG Calls, that's JAG Calls, out at jagcalls.com. Hey, and while you're at checkout, use OD 2024 for a discount. Once again, OD 2024 for your discount.
Now let's get back to today's episode.
So, let's play like a what if situation. Kind of like you said it was Minnesota, right? That it pretty much wiped out their whole deer population.
Oh no, no, that would be real bad. No, in Wisconsin,
Wisconsin, okay, the cheeseheads.
Wisconsin, where they have in sort of their core area, there's sort of four-ish counties, certain areas in those counties, they're seeing population decline. So there's still deer there, and people are still deer hunting, but when you look at the population trajectory, it's tipped down now, because they've sort of gone past. If you see, I want to say it's like 23% prevalence in does, that's when they start seeing population declines. And they're seeing the same sort of thing in Arkansas, in Pennsylvania, where they've had the disease for a long time. So it's being replicated in other places. And years ago, they did the same sort of research in Wyoming in mule deer and whitetails and saw population declines from CWD.
So, okay. With that being said, so Oklahoma, we can use feeders. We have pretty much clay soil. And I don't know, I'd have to say at least 80% of the state. So say if we get a bad outbreak, what do you think, or what could you see happening here? Worst case.
Yeah, well, I mean, it's sort of the same thing happens everywhere. It's just the speed. So what you're talking about, with the soil that just, holds prions, so the exposure to deer could be higher. It would really hinge on what, how many deer are out there on the landscape. Like some of these areas in Wisconsin and Pennsylvania had super high deer densities, like, 30, 40 deer per square mile. So ideally, if the deer density isn't that high, then the transmission would be slower. But then you talk about feeding brings animals together, nose to nose. They can leave their snot and everything else behind at the feeder, around the feeder. So that just increases the likelihood of encountering a prion then, if you're sort of artificially congregating animals. I mean, it's like, during COVID, we didn't go out and, nobody was going to bars and like hanging out in big groups then, because we know that increased the transmission. So, you're just inviting everybody to a big contaminated salad bar where nobody's, using the sneeze guard or the tongs, and they're just stuffing their faces right in the salad bar. Like, would you want to eat at that?
I was going to ask, where are we at with like detection methods? Like, I know it goes state by state with how they, do it, but where do you think we are as a whole on detection methods?
Yeah, so the testing has not progressed that fast, and that I think is frustrating to a lot of people. So the two major ways to test are, the first one is called immunohistochemistry, and basically that's where they take tissue slides and stain them, and if they light up when they attach to the prions, then you've got a positive. So that's sort of the confirmation test, but it's sort of slow. It requires a pathologist, like a trained person, to read those slides, so that slows things down too. The test that most states use during hunting season, when we get the majority of our samples, is called ELISA, and that's just an acronym for an enzyme test. And that just, you take a piece of either the lymph node, so we use the lymph nodes that are kind of behind the chin here, or a piece of the obex. And those samples aren't easy to get for most hunters. Like you have to be trained where they are. And we've had good success with training taxidermists to collect lymph nodes, because they're down there digging in the head and everything. So they've figured out those tissues pretty well. But you have to take little chunks of the lymph nodes and then run them through this ELISA. And the test takes about 2 days to run. There are other tests that are used now more in research settings, and we're trying to get some of those approved by the USDA so states can run them, that are a little bit more sensitive, but they also have the higher potential to throw false positives and false negatives. So there's some trade-offs on all these. And of course, every hunter, wants like a dipstick to be able to test in the field, especially, in an area that's known to have CWD, because that's a problem if you take that deer to a processor and you pay, whatever processing fees are now, $100+ to get that deer cut up, and then find out, 2 weeks later that it's positive. It's also a problem if you take it to the processor and the processor cuts it up, grinds it up, and mixes meat from other people's animals in, and you didn't shoot a CWD positive deer, but the bratwurst you had made contained some prions that you were unaware of. So yeah, having faster tests would definitely be helpful. But because this is a protein, it's not something we see often. So there are people working on, fancy test development that I don't entirely understand, that I hope are better. And I hope that we can use samples that are easier for hunters to collect. But, what was it? It was 5+ years ago. I asked some of the best researchers. I happened to be sitting at a conference with them, the people who develop these tests. And I said, are we going to have like a carcass side test in the next 5 years? And they said no. And that was 5 years ago. And so we haven't seen that yet. There's some technologies you can take into the field, but they're not ones that like hunters could do themselves.
Mm-hmm. So if there's an outbreak, I see in Oklahoma, like they kind of like they put restrictions in the area. What's the best thing to do if there's an outbreak in an area or we detect it?
Honestly, the best thing to do, if you find, a very, like just a few cases at the very beginning, you think you're at the very beginning of the outbreak, kill all the deer in that little area, where you think that first index animal was, to figure out, how bad it is. And then 2, if that was, the first introduction, you can hopefully stop it. But what we see is, the public is like, oh, we don't want you to kill the deer, but they don't realize it's either the deer are going to die now, or you're going to have this long protracted disease that's much worse over time. So if you can support that, give the agency 3 years to just hit it hard right then. And then if they can't get the job done in 3 years, say, okay, we're going to move to a new strategy. The best thing that hunters can do actively, besides, supporting this response activity, is to go out and hunt, take deer, take antlerless deer, fill every tag, get extra tags, try to decrease that local deer density, because that's going to decrease transmission. So everybody, I hear it a lot that people think that CWD is going to ruin hunting. Like, I think people should hunt more. I think, there's tags out there to be had. You should fill them. And I'd like to see more state agencies. There's some people that are Uber hunters out there. Like, let those people go out and, fill every tag, donate the deer, whatever they can do to kind of get that density down. Because we need to stop that transmission process, that deer to deer, before they really start shedding it, in their fluids and contaminating that environment. So those are the best options, I think. Sort of short term. Long term, it gets really tricky. And, then you're just talking about trying to contain the spread, trying to not increase the prevalence too fast. And that's when you get into regulations about, not moving carcasses, deboning, if you're traveling somewhere to hunt, make sure that you debone the carcass, that you don't bring back, like the brain and spinal cord have the most prions in them. So if like you're going to do a European mount or something, clean the brain out where you are, rather than like bringing it home and power washing it, because you might just be introducing prions to like your backyard, which obviously you don't want to do.
Yeah, there's like nothing you can do about it. I feel like if it's there, it's there. That's what's scary.
Well, that's, I was thinking about this question, because a lot of people just kind of throw up their hands and like, oh, there's nothing we can do. And if you look at it sort of on like a macro scale, yeah, it seems like overwhelming. And there's, 36 states now that have detected CWD. But there are places, like I said, where the state agency goes in and hits them hard. Like New York did it in 2005. Minnesota's done it. Missouri's done it multiple times, like 3 or 4 times, where they had a detection and they went in and really, decrease that local population just right around where the detection was. We're not talking like the whole state or whole county or anything.
Right.
And they haven't detected it since. So
Wow.
I think that story needs to be told more. And people, if they can be patient and just say, okay, give them 3 years to just knock it down. And it, like I said, small area, you need it to be focused. This is like a surgical strike. This isn't, a carpet bombing of all the deer.
I think that was part of the. What do you think went wrong with the Wisconsin deal as a whole? You think it was just mostly miscommunication? Was it, because it's been a while since I've heard that case study, but
Yeah,
what do you think went wrong most with that ordeal?
that's a good question. I think I probably would have been able to answer this one better a few years ago, but I've, been away from it for a while. I think that the public, like you say, doesn't really necessarily understand. Like they think EHD is bad because they see dead deer. They don't necessarily see the dead deer, especially at the beginning of a CWD outbreak. So like everything looks fine. Why are people freaking out? Like, why are you ruining my hunting? And it's not necessary, if they're liberalizing tags and everything, they really wanted people to take antlerless deer, but our hunting culture is sort of so ingrained now. And I am a hunter now. I should have clarified that at the beginning. Like I started hunting in Wisconsin in this hot zone. So I don't think it should be something where we're like, oh, CWD is killing hunting. Like that's not happening yet. And what we worry about when we talk about these population declines, like we're not over that hill yet. Like we're still climbing up the hill, and maybe some of these places are getting to that top and might be peaking over. So I think that story has yet to be written. But going back to what you were saying earlier, with antlers, the hunting culture is still so reticent to shoot does that, oh, we have to protect does. We don't want to decrease the density. Hunters want to see deer, obviously, because that makes them hunt more enjoyable. Like sitting in your tree stand, being bored, not seeing anything isn't super fun. But thinking about, the long-term outlook, like I used to talk about CWD as being a problem you needed to think about for your grandkids. But what I've seen over the past few years, is that it's speeding up. Like we've kind of got that exponential curve where the disease is getting worse faster. And so I've got a 12-year-old son, and he just passed his hunter ed this year. Like he sat in the stand with me last year. Like I worry about him hunting in a few years, like what that means for him, just because it's going so much faster. And with people, if you travel to hunt and you bring back stuff and don't know the proper way to dispose of it. So, the role that hunters play in moving disease and how we need to take some ownership of, our role in these actions. Cause it's not just deer, trotting across the country, spreading prions, we are taking it down the highway pretty fast.
And when you say dispose, like, how do you dispose one properly? Because I feel like once again, I just don't feel like there's like a right way or unless I don't know. So
Yeah, so it depends on what you're doing. So like if you have your own property and you're hunting on your own property, and you shoot a deer there, like you can dispose of the gut pile and carcass and everything on your property, like the deer died. But if you're traveling somewhere and it's, obvious, if it's a known CWD area, they should give you guidance on how to dispose of things. A lot of places are offering dumpsters now. So you can put, all the waste, gut pile, bones, everything in those dumpsters, because those carcasses have prions in them. But it's when people, they shoot a deer and they say, I don't want to, mess with it now. They might field dress it and then throw it in the back of the truck and drive home, however many hours. And then they get home and cut it up there. And they say, oh, well, I'll put the scraps out, for the scavengers. I'm doing a nice thing. That could introduce prions to a new area. Or, they shoot a big buck and they say, okay, I'm going to boil the skull. And the water, obviously is nasty. And so they go and dump that out in a field. That introduces prions. So if you are moving an animal, right now it's mostly about states. A lot of states don't let you bring in whole carcasses. They want the carcass to be deboned. And so, I think hunters have a responsibility to learn how to debone their carcasses. That, it takes time. If you shoot it at dark, you track it and you're tired and you don't want to deal with it, that's hard. I get it. We're asking hunters to do more stuff. And hunting's already hard enough. But if you are somewhere, you're going to move that carcass, leave all the parts that you don't want to eat or that you're not going to mount in the trash somewhere, bag it up, make sure that it ends up in a landfill, that it doesn't just go in a bone pile or a pit or something like that, where scavengers or other wild animals could get to it. That's the best option, that these products end up in a landfill.
Okay, that's what I was wondering. I was like, where do they need to go to where? Because nothing in my mind was kind of where would be.
I realized I said dumpster, but then I didn't say where the dumpster would end up. So yeah, landfill is the best option.
I guess that's probably because least amount of animals are probably in a landfill digging through after they're buried versus just laying on the surface somewhere. That makes sense.
Yeah, and most landfills are lined now, so that helps keep the prions contained right there.
Mm-hmm. And how long can prions live in the soil? I don't know if you went over that.
There was a study done on sheep scrapie prions, and the researcher actually put infected brains in soil in flower pots, buried them in the backyard, dug them up 16 years later, and they were still infectious. So that's sort of been the longest anybody's been willing to wait. There were studies done
so far.
Yeah, that's a patient person. There were studies done at the research facility in Colorado where deer died of CWD. They left the carcass in the pens, waited 2 years, put deer back in those pens, and the deer got infected. So these things, like you were talking about the person with the elk with feeders and trying to disinfect them, to disinfect equipment, if you're cutting up a deer, you want to use a bleach solution, like a 10% bleach solution on your knives, because that actually denatures the prions. So they're really, really difficult to get rid of. So you want to soak those that stuff in bleach, not straight bleach, because that'll eat away your knife, but like a 10% bleach solution, and just let it soak for an hour to get rid of prions.
Does heat get rid of prions at all?
Really, really high heat, not like a nice medium rare isn't going to do it. We're talking incineration temperatures, 1600 degrees Celsius.
Oh, wow.
Yeah, more heat gets rid of more, but if you want to get completely done with it, it's charred.
Wow.
So I guess I'll ask the big bad question. How nervous should people be about a crossover into the, how nervous do you think people should be?
I struggle with this one a lot, because I think there's evidence on both sides. So the mad cow outbreak, obviously is sort of our best example. So with that, they noticed people started getting infected with it. And it wasn't a ton of people. You think there were lots of cows put in the food stream, and like 224, I think, worldwide got infected. So it was just like a subset of people that probably ate contaminated beef. But they noticed that they were younger, like in their 20s and stuff. They started having gait abnormalities. Their eyes weren't tracking right. They had speech issues, and then they eventually died. So that's the scary part. But on the flip side, we have scrapie in sheep, and people have been eating scrapie infected sheep for hundreds of years. Like scrapie's been around for a long time, and nobody has been known to ever get that. So I guess I'm worried because I feel like we're running a natural experiment right now where more and more people are going like, nah, I'm going to eat it anyway, which scares me a lot. Like I would not eat a deer with CWD. Like if I knew that deer had CWD, like that thing's going in the trash. Well, first I would call the Natural Resource Agency, because a lot of times they'll take it and they'll incinerate it for you. But if they tell me to put it in the trash, I would dispose of it, even if I'd paid a processor and all that. Like no way. But I don't think there's a high likelihood that it would jump to people. I think there might be, kind of like with mad cow, a small subset of people that might, maybe their genetics makes them likely to have abnormal prions, because people do get these transmissible spongiform encephalopathies. Like naturally, there's a spontaneous form called Creutzfeldt-Jakob disease that happens about 1 in a million. There's all these crazy names for other genetic diseases, like Gerstmann-Straussler-Schenkler syndrome. There's fatal familial insomnia, like all these genetic diseases that were passed down through families. So, people can get them, but with CWD, I don't even know that it's as much of an issue about if a person actually got it, just even the chance that a person got it. You might have heard about, recently it was out of some med school in Texas saying, oh yeah, 2 people had CWD, 2 hunters, which turned out to be totally false. But it was a story that was picked up by Field and Stream. And I spent so much time trying to combat that story because it was just based on a medical student's poster that somebody kind of took out of context, and there was no evidence for it whatsoever. But yeah, it's hard because even the perception that people could get CWD, I don't want people to stop hunting. Like I don't want the public to view deer as like a menace or a threat to their health. That would be terrible. They're a national icon. So I really don't want that part to happen. And that's what I'm worried about, that we might get to that point where people are really worried about deer affecting them and don't want them around anymore.
So right now, what studies are you working on at Cornell? I mean, I get CWD, but like kind of what?
Well, I guess, yeah, the big one on CWD, I'll mention quickly, is called the Surveillance Optimization Project, because we were seeing that all these states, are doing CWD surveillance, and they spend a lot of money on it, but they're all doing it a little bit differently. And so you can't compare across states and say like, okay, this state has a prevalence of, whatever, and this state hasn't found CWD yet. Is it because they haven't found it because they're looking, or are they just not testing enough animals? So we realized, a lot of times it's like you talk to Dallas, and it's the deer biologist for the state who's already like way overtasked with their job anyway, and they become the de facto CWD expert. And so they're trying to do their regular deer biologist job and plus deal with CWD. And so they don't have the bandwidth a lot of times to do all the stuff that CWD surveillance might need. So we developed this project to try to help out states, provinces, tribes, by giving them tools, a way to manage their data, a way to run models, like complicated models, but run it simply where, you can click a button, or, use a slider to kind of see different scenarios and inform yourself on what kind of surveillance you should do. I actually developed this model called the hazard model, and that we developed it for New York. And I helped Tennessee, and we ran it in Tennessee, and that's how Tennessee found CWD. After that, Georgia, Alabama, and South Carolina all came to me, and they're like, "Can you do the same thing for us?" I'm like, "Really? You want to find CWD?" And then Alabama and Florida found it, and now Georgia. So yeah, trying to give states tools is sort of like one of my biggest areas of research, and seeing what we can do, with things like machine learning and whatnot to try to advance our knowledge across a larger landscape. That like each state kind of doing their own thing isn't advancing our learning as fast as we could if we have larger data sets and people working together. So yeah, that's a big one that I think is really useful. As far as other research projects, we wrapped up a project on moose recently that was pretty cool. Like did helicopter captures and caught moose, and found a lot of them were dying from giant liver fluke, especially younger ones. So we have a very small population in the Adirondack region in New York. And, everybody thinks about brainworm because interaction between white-tailed deer and moose can introduce brainworm to moose, and they don't do well with that. But we also have giant liver fluke that was causing a lot of problems. We didn't have winter tick, which is, really big issue for states like Maine and Minnesota, where, thousands and thousands of ticks get on these moose. Let's see, what else? I have a student working on lead issues in things like Fisher. We just talked about reproduction in pine marten today. So that is pretty cool, trying to look at mass cycles and small mammal prey availability and reproduction in pine marten. So I have a pathology resident that was cutting up teeny tiny ovaries from these pine marten. There's some people working on different amphibian projects. So we had projects going on hellbenders, like the giant salamanders for a while. So those are pretty cool, but doing things with like eDNA, environmental DNA, to try to find species and pathogens. So yeah, I could rattle on for a while.
What if you had to pick, like, which I know it's probably gonna be hard for you, but what would be like your maybe top 3 funnest studies you've done?
Well, I used to get to go in the field and actually like do things, and now it doesn't seem to, I'm the one like making sure that the students can go and do things. So yeah, I mean, doing my PhD work in Wind Cave National Park was pretty awesome. I got to be in the helicopter. I darted deer, I trapped deer. I went out with other grad students, so we're working on mountain lions and coyotes and pronghorn. So we're all kind of out there together. That was probably the best time. With the moose work, that was mostly the helicopter crew got to do that. So they would just kind of come up back and like hand us the samples. So
Yeah.
Not as much fun there. But yeah, so I don't know. I mean, I feel very fortunate in this job that I do get to work with really cool species. And I kind of see all the health work we do as being like a value added. Like it doesn't necessarily have to be the reason that you're doing the research, but if you got hands on the animals, you might as well take the samples while you've got them.
Right.
I've been lucky. I got to do prairie dog research in the Thunder Basin, and then a loggerhead shrike. So it was like same 2 different projects, but I was there for the same. So in the early morning, we do point counts for the prairie dogs, for the pups and the adults, and then we'd also do bird counts. So I got to do that, and then I almost got to do pronghorn fawn capture. I didn't. I missed that one by like 2 days.
Yeah.
I missed that by about 2 days up there. But
bummer.
then I got to be a part of one of the first glade projects in Cookson. Like they kind of they're starting to do projects on the glades. They're like just basically taking basic inventory
Mhm.
of amphibian and reptile species up there, and insects, just seeing what's there, what utilizes it, and then also bird counts on and off glade and stuff like that. So yeah, basically I was walking around looking for timber rattlesnakes, so that was fun.
I felt like you were successful at that. And then you also, while you're doing the prairie dog, you got to see was it a western or a prairie
Swift
rattler
Swift
going in?
fox.
No, the rattlesnake that was actually going down in the hole, in the prairie dog hole.
Yeah,
You sent
that
me
was a prairie rattler, and that was over in the Rita Blanca.
Yeah, that was neat.
Yeah, those are
But
cool. I'm trying to get better. Snakes are still one of my species that I'm sort of like I when I was cruising around in South Dakota, I saw a snake and I swear I levitated for like a few seconds, just like
Yeah,
straight up in
it's
the
like
air. But I got to work on bats in Wisconsin when white nose syndrome was first hitting, and it's one of those things like when you really get to work with an animal and see it up close and sort of appreciate it. Like, so I'm a big fan of bats now because they were just so cool. Like they were little faces and they're so tiny and they were just really pissed off at you and just biting the crap out of you and trying, like they weren't intimidated at all and not cowering. They were like, I'm gonna get you and I'm gonna rip you
yeah,
to shreds.
yeah. I definitely messed up on my degree. I wish I've done something more like you guys did because
did you
I
do
had
Did you do the bird mist netting? Were they doing it then?
They weren't
Or
doing it that I'm terrible at birds. Like I should be better. Like
yeah,
when I hear people like, say they're doing point counts and identifying bird songs, and I really shouldn't admit this because the Cornell Lab of Ornithology is like a big deal.
yeah.
And I feel like I should be a better birder just because it's like, the proximity to it. So, but yeah, it's sort of interesting. I feel like people, especially at OSU with like, the wildlife degree and everything, have such a better sort of natural resource background. Like I took all sort of the regular old biology classes like anatomy and physiology, which are good now for the field that I'm in, but I had to teach myself a lot of, the regular, ornithology, mammalogy type stuff. Um, I took human dimensions when I was finishing my PhD because I knew that was something I needed to have, but hadn't ever been offered it throughout. So yeah, it's definitely a cool field to be in.
yeah.
I'll tell you,
I messed up.
cardinals are mean.
Cardinals are mean.
They're very mean.
It's my
You do,
favorite
yeah.
bird.
Just be prepared now.
Favorite bird too.
If you're mist netting those suckers, just be prepared. When you grab it, he's gonna just go to town.
Uh-huh.
And he's not afraid of you at all. He's like, I'm taking a piece with me.
Yep.
That's what he says.
Yeah.
So
It's like I'm red for a reason. This means warning. Stay back.
yes.
I like cardinals and hummingbirds. I am
Mhm.
fascinated with hummingbirds. So I
Yeah,
would
they're
love. I always look at their like the ways they travel and how fast they can go. That stuff's so fascinating to me.
I know. Like you think about like people building jets or helicopters or whatever, and none of them are ever going to be as good as a hummingbird.
Yeah, and how little they are. I've
Mm-hmm.
always been amazed how they can survive our rain and hailstorms here. I mean, I know some of them get hit, but still, I'm like that little tiny animal is a warrior. I'm in my house hiding, and they're outside taking it on, and they're the size of my pinky. So,
Exactly. You got to be tough to live outside. That's why
yeah.
we are wimps and build houses.
So, Krista, for anyone that's listening and wants to find out some of your research more in detail, where can they go to look up this?
The best place would be to go to the Cornell Wildlife Health Lab website, so they can check out all the cool projects we're doing, and we've got a bunch of different disease fact sheets if they want to look anything up or see about our research projects.
Okay. Well, thank you for coming on today's episode. We enjoyed talking about CWD. Anyone listening, that link will be in the description below on any of the platforms you're listening on, so definitely look up that. But thank you for coming on today, Kristen, and talking about CWD with us.
Absolutely happy to do it. Thanks.